Assalamu alaikum and welcome to Learn with Laraib. Today we will be discussing GIT secretions. So we'll go with the flow of the food. Once it reaches the mouth, the salivary glands secrete saliva, which consists of salivary amylase and lingual lipase, also mucus. The composition of saliva is hypotonic. It has increased potassium and bicarbonate. However, it has decreased sodium and chloride. Now, once the bolus reaches the stomach, let me show you the diagram of stomach first. We have the fundus, body, antrum pylorus, and cardia. So, when it reaches the stomach, G cells present in the antrum of the stomach release gastrin, which then releases gastric acid from parietal cells. Parietal cells also secrete intrinsic factor, which is involved in absorption of vitamin B12. Now, the main function of gastric acid are three functions. So it includes killing the bacteria. It also denatures the protein. And the very main function is that it converts pepsinogen released from chief cell into pepsin. So as you can see in the green, I have drawn the cells in the fundus and the body. These are the chief cells. The G cells, I've drawn that by mistake in the fundus and the body. They're present in the antrum. So mostly the chief cells are present in the fundus as well as the parietal cells, and they're also present in the body. Now coming to another hormone release, which is somatostatin. It is an inhibitory hormone and it inhibits the gastrin, HCl, and histamine release. Another function of gastric acid is that it stimulates enterochromaffin-like cells to produce histamine, which in turn stimulates the parietal cells to release gastric acid. So it contributes in release of gastric acid. Now the chyme made by the stomach, mostly churned by the pylorus, then moves to the duodenum. Here's a diagram of duodenum and a leaf-like pancreas and gallbladder. This is not accurate to scale, but this is okay for demonstration. Now when the partially digested chyme moves into the duodenum, the fat specially stimulates the eye cell to release CCK, which then in turn stimulates the pancreas to release digestive enzymes, which are lipase, amylase, and protease. Here you can see CCK signaling the gallbladder and the pancreas. And for gallbladder, it initiates the contraction. It also relaxes the sphincter of Oddi to allow bile and pancreatic juice entry. Also delays the gastric emptying so the digestion in small intestine can proceed efficiently. At the same time, the S cells in the duodenum sense acidic chyme and secrete secretin. The secretin stimulates the pancreatic ductal cells to release bicarbonate and increase bile secretion, not from the gallbladder, from the liver. As you can see, the S cell signaling the pancreas and the gallbladder. So this makes the alkaline environment suitable for enzyme activity since the pH is now corrected. Secretin now also inhibits further gastric acid production. As I said, the main trigger for CCK is fats and protein mainly. Now glucose is left. Glucose has to do something as well. So when carbohydrates arrive, the K cell of the duodenum and jejunum, as you can see in the diagram, secrete glucose-dependent insulinotropic peptide, which is GIP. This enhances insulin release from pancreas to aid glucose utilization and simultaneously inhibits gastric acid secretion. Alongside the enterochromaffin cells in the intestine secrete serotonin. So in the stomach, the ECL cells secreted histamine. Now its brother serotonin is secreted, which increases the motility. Also an important component, VIP, vasoactive peptide, is secreted from parasympathetic ganglia, which enhances water and electrolyte reabsorption, as well as it relaxes the smooth muscles. Also, it is involved in saliva release. Also, one point I would like to mention in saliva that it contains an enzyme lactoferrin, which inhibits bacteria's action. So now, as I said, these cells are scattered through the stomach, intestine, and pancreas. So somatostatin inhibited gastric acid production, and it will also inhibit the pancreatic enzyme production, the insulin and glucagon release, as well as bile acid secretion. So to conclude, the bile control is via CCK and vagal stimulation via acetylcholine. Both of these contract the gallbladder for bile release. So the gastric acid production is inhibited by the K cells as well as secretin. So this was all for GIT secretions. You can take a screenshot from the screen. And for more such videos, subscribe to my channel. Thank you.